We have a really exciting event today on the topic of the origin of life and self-organizing systems with three fantastic speakers. For those of you that are new here, and as a reminder to those that aren't, we ask speakers to prepare a typical 1 hour long talk, but we allocate 90 minutes for each speaker. This is meant to encourage you, the audience, to ask lots of questions and spark discussion. And in person, these events are quite lively, but obviously it's hard to capture that sort of dynamic in a virtual format. So we found it works best to have the audience type questions into the chat, and we ask the speaker to periodically pause and check for questions. If you have a problem with that and feel you really need to ask your question out loud, please message me or Gwen. We also have a 30-minute break between each talk, so you have a chance to stretch your legs, grab a cup of coffee or a bite to eat. So the talks are at 11, 1 p.m. and 3 p.m. today. So with that, I'll get out of the way. Our first speaker of the day is Sara Walker from Arizona State, who will tell us about life is what? Sara, welcome.
Excellent. Thanks so much for having me. I'm very thrilled to be here. And I broke my talk out into 3 parts because I had the impression that you do different sort of formats. So the first part, I'll talk for about 20 minutes and then maybe take a 10-minute break. And then there's like 3 parts structured like that. But feel free at any point to interrupt me because I'm happy for this to be more of a dialogue. I know that's kind of the style that you would like to encourage. And that's also more fun for me because I think there's a lot of ideas I'm hoping to introduce in the talk. And some of them may be rather provocative. So it's good to have some discussion about them. So my background and training is in theoretical physics, but I work mostly in astrobiology now, which is primarily focused on the origins of life and the search for life in the universe. And I'm deeply interested in whether there are universal laws that govern living systems that would apply both here on Earth and also in other planetary environments. So that's sort of the core motivation of my work. But if you're working sort of deeply on this question about what life is, you can't but help try to dig at sort of some of the conceptual foundations of the issue. So what I wanted to do in the first part of the talk is it's not a technical part, it's more philosophical and more dealing with the concepts about how do we approach defining life, or how should we think about building theories of life, and what are the challenges that we've faced so far versus where are the kind of directions that, at least I think from my perspective, we need to go, which may or may not be the right framework. But I think reframing the question is critically important to address some of these hard questions that we've been confronted with, at least for a century in origins of life science, if not millennia, if you think about how old, the question on the nature of life is. So the first part of my talk is really on this question, what is life? And it's not for lack of trying to come up with definitions for life. We have lots of them. And sometimes I'll put up a word cloud with all the different definitions. There's lots of overlap between different ideas that people have proposed. But I really like this really short paper by Carl Sagan, where he digs into some of sort of the most popular definitions. And this is a definition that you might see, for example, in your biology textbook, where life is defined as any system capable of performing a number of such functions as eating, metabolizing, excreting, breathing, moving, growing, reproducing, and being responsive to external stimuli. So it seems pretty straightforward, right? You can, these are all things that life does. But what Carl did in this paper was he basically pointed out that if you take this definition, you get some very non-intuitive examples. And for example, if aliens were visiting Earth, they might think that cars are the dominant life form because they have all of these functions, and if that was their definition for life, they should assume that cars are life. And in fact, cars have also been to space, so there's some more evidence there that they might be a life form. And so he ended up, coming to this kind of conclusion that the most attempts to define life come up with these kind of counterintuitive conclusions. So many properties are either present in machines that nobody is willing to call alive, or absent from organisms that everyone is willing to call alive. So his point was there's really no viable definition, and that still stands today. So even though this paper was written several decades ago, I actually disagree on this point about machines, which I'll get to shortly, and why I include them sort of in what I think of as living physics. But I think the point that he was making with this analogy is actually a little bit deeper, which is if you start with an attempt to define life and you try to think about the things you want to include and the things you want to exclude, and then you proceed axiomatically from there to construct the definition, you always end up with these counterintuitive cases where something you meant to exclude is included in your definition, and something you intended to include is excluded. So it's very clear you can't draw a hard boundary around what we consider to be life, and come up with a very rigorous definition just by doing this kind of approach of just looking at the structures that we see in the world and trying to define their properties and say that's what life is. And so this is not, you know, an unhotly debated issue. So some of my chemistry colleagues in Origins of Life have really taken issue with a lot of definitions of life, or spending so much time attempting to define life. So when I was a postdoc, you know, really starting to get into the origin of life field, I went to the American Chemical Society meeting, and I was rather shocked to see Andrew Ellington, who's an RNA chemist that works on RNA World type things, say that life does not exist. To me, that was kind of like very deeply insulting. I was like, surely I'm sitting here and I must be life. But I think he was making actually a really deep point, which was better articulated in some ways in a paper on defining life by Jacques Shostak, where he was basically arguing that definitions of life don't help to solve the origin of life, where he points out that as one focuses experimentally on any of the defining properties of life, the sharp boundary seems to blur, splitting into finer and finer subdivisions. And so basically the point is, which is something that, you know, we all know, a molecule is not alive, a molecule is not life. So when we look down at the components of life, we don't see the properties that we associate with life. Life is a system level or emergent property. And this is one of the reasons that it's been really hard to define. But we have some intuition, or at least those of us that are trained in physics, that there should be some principles underlying living systems, even if you can't talk about those principles in terms of the component parts. And so I really like this quote from Einstein, who, you know, periodically in his writings would talk about this dream of a vision of including life in the laws of physics. And this was in an address that he gave on the occasion of Max Planck's 60th birthday, where he says the general laws on which the structure of theoretical physics is based claim to be valid for any natural phenomena whatsoever. With them, it ought to be possible to arrive at a description or a theory of every natural process, including life. So Einstein certainly had this view that life should somehow be accounted by laws of physics at some point in the future. And I think most of us that study physics hope for that to be the case. But it's certainly not a universally shared view. So just to offer a contrasting viewpoint, Stuart Kauffman, who's a theoretical biologist and has thought very deeply about origins of life from a theoretical perspective for decades, makes this argument that what life is doing, whatever it is beyond physics. And there is no law that you could write that would explain the evolution of the biosphere. And part of his argument there is that life generates novelty and you actually can't predict what structures are going to be formed. And so there's no possibility of writing down a closed form mathematical equation to describe that process. So there's a lot of debate in the community. I tend to be on the side where I think there are laws because I am biased by my training as a physicist and romanticized by the vision of simple explanations for physical phenomena. But I think that we have to take very seriously this counter viewpoint and the fact that we haven't been able to accomplish that yet, that it might not be possible. But I think we still need to do it, especially in astrobiology, because if we're talking about trying to look for life on other worlds, we have to have some sense of a universal property of those systems that we're looking for. So this question, what is life?
Sara, can I say?
Absolutely.
Is it clear what physics is? If you're
No.
getting into this kind of detail, or okay.
No, so I'm actually, in a few slides, I'm going to kind of question some of the foundations of physics. So my personal viewpoint is actually the approach, like the mindset of the theoretical physicist is probably a good mindset to have about trying to address the problem of origins of life because of our ability or like our training to be able to build abstract principles. But I don't think that those principles or those approaches are going to look like the way we've done physics before. So it depends on what you call physics. You might still, I mean, and what I call physics is sort of like from Newton on, because Newton invented modern theoretical physics. And most of us, when we do theoretical physics, do it in sort of a Newtonian style, which I'll talk about that in a minute. But this is a really good question because it's kind of challenging the boundaries of what we consider to be disciplines, right? And disciplines themselves evolve over time. So maybe what we're talking about is a discipline that hasn't emerged yet. Sorry, go ahead. I can't see who's talking. So.
Hi, this is Vadim Oganessian. I'm not even a newcomer to this field. I'm a physicist. I don't think about life. But I was wondering if you have a set of and I was a little bit late, but you seem to be concerned that certain criteria about definitions of life readily produce counterexamples that clearly indicate insufficiency of these
Yep.
criteria. But so in physics, when we want to sweep counterexamples under the table, well, under the rug, we can think about applying principle or computing things that are statistically significant.
Yes.
Right. So one or two counterexamples are not going to throw off our confidence. So to say, has anybody actually tried to think of life statistically, that certain things is,
Yeah, I will actually, the second arc of the talk is really going to be about building a statistical approach to characterizing biochemistry. So you
Okay,
could talk
thanks.
about, like, from the perspective you're talking about, like, one of the ideas I had there is you would have a likelihood distribution of what you expected to be the statistical properties of biochemical systems, and you might have some outliers, and then you just wouldn't know what to do with them. But the defining life problem is even worse than what you're saying, because it's not even like you have a quantifiable criteria in the first place. They're just descriptive definitions. They're not theories. They're not quantitative frameworks. And so when I got into origins of life, as a PhD student, that was sort of the first thing that was rather shocking for me, is that nobody in the field seemed to be working on whether life was a quantifiable phenomena, and you could talk about the origin of life as a transition where you could actually have a mathematical measure that would tell you this system was less alive, this system was more alive. And so that's very much the perspective that I approach the problem. One more point on this, which I was going to get to a little later, but I can just say now because it's really related, is part of the issue. People go in to try to define life. And what you really want to do is try to understand sort of what are the underlying principles of the universe that we haven't really identified yet, that become the explanatory framework from which you can derive the properties we associate with life. So I think part of the problem is we're trying to define this phenomena that we see in our subjective experience, and the objective description is not exactly mapping to that phenomena. I'll get to some examples of what I mean by that in a minute, at least from the history of physics. So anyway, just are there any questions at this, other questions at this stage? I think some of the things I'll go through will kind of clarify my perspective on it, and then we can have some more discussion on it. But I think all of these points raised are really excellent. So this is super fun.
So can I ask a quick question?
Absolutely.
This is David Nelson. Could some of this be related to Phil Anderson's ideas about more is different?
Yes, absolutely.
Okay.
I mean, like many people, I was very inspired by his writing on that. So I think I did have a little bit different perspective, because I actually visited Princeton when I was a postdoc and gave a talk, and he was in the audience, and he was very energy focused in origins of life, and very much in sort of the Stuart Kaufman camp as far as his thinking on that problem. And I'm very much that the origin of life problem is an information problem, not an energy problem. But there were some philosophical differences about what the more was. But not deeply so. I think that there might have been some agreement there.
Thank you.
Sure thing. But yeah, that stuff's very inspiring. So what I want to, so anyway, I just wanted to get into the point of introducing at least Schrodinger, because there is a long history, obviously, of people asking from a more like physicist perspective, more fundamental perspective, what is life? And probably the most famous treatise on that was written by Erwin Schrodinger, who's obviously famous for his work in quantum mechanics. And he gave this series of lectures in 1943 at the Dublin Institute for Advanced Study, which he turned into a book called What is Life, which is fantastic. But in that book, he asked, how can the events in space and time which take place within the spatial boundary of a living organism be accounted for by physics and chemistry? So he's very much interested in whether there was a physical, chemical explanation for life. I've highlighted within the spatial boundary of a living organism because I think that's actually the wrong framing of the problem. And part of the thing I hope to convince you today is that if we're not asking the right questions, we're not going to get the right answers. And whether or not we've gotten to the right questions or not yet is one of the things that I think is really deeply intriguing about this field, because I don't think that we know how to ask this question properly yet. So this kind of brings me to the title of my talk, which is the life is what part. In part, that's kind of meant to really indicate this idea that I think we don't know what life is, and I think we're going to be deeply surprised by the answer. So when we find out the answer, we're going to be like, life is what? What is it? Oh my God, that's really, really surprising. But I think that that's actually, also in line with a lot of people that have thought very deeply about this problem. So for example, Schrodinger in his book, although he was able to make many really important predictions and statements about life from the perspective of physics, including predicting the structure of DNA. He admitted at the end of the book that probably life, while not eluding the laws of physics as established up to date, is likely to involve other laws of physics, hitherto unknown. And so to me, this is sort of like a major motivation in my work, is I really do think that new principles are going to be necessary for a variety of reasons, more reasons than I'll have time to cover in this talk, but I'm going to try to go through a few of them now. One of them is that we obviously have a very beautiful history of understanding the physical world when we think about what we've been able to accomplish in fundamental physics over the last 350 years since it was invented. But there's this kind of critical point that's missing, which I think is nicely captured in this quote from David Krakauer, who's the president of the Santa Fe Institute, that the theory of everything is a theory of everything except those things that theorize. So physics as we know it so far is absolutely brilliant and elegant at describing the physical world, but it doesn't describe things like us. So this raises some questions for me, at least, about the foundations of physics and how we should be thinking about this problem. And I just have actually I gave a talk recently about like sort of all the problems with foundational physics that need to be solved in order to get at the origins of life. But one of the interesting things is that the equations of physics were invented by people. And so I really like this quote from Stephen Hawking in A Brief History of Time about, even if there's only one possible unified theory, it's just a set of rules and equations. So we have to ask, what is it that breathes fire in the equations and makes a universe for them to describe? So there is this deep problem in physics as it's structured now about we don't know where the laws of physics themselves come from. And Frank Wilczek has this really fabulous paper up on the archive called Physics in 100 Years, where he goes through, from the perspective of a theoretical physicist, all the things that might need refinement and be developed in the next 100 years of development of physics. And one of the things that he points out, which is something that has been kind of hounded into me through my career from my postdoc mentor, Paul Davies, is that we've kind of inherited a legacy from Isaac Newton about thinking about the laws of physics in terms of dynamical laws that paradoxically live outside of time and aren't described by the physics themselves, and initial conditions on which those laws act, which in turn have to be specified by the user, i.e. us, and potentially fine-tuned to describe the world that we have. So I don't necessarily want to go into all the details of why I think this framework is maybe not the right one for life. Some of those ideas might come out in my talk, but the main point I wanted to say is the laws of physics as they are designed now were developed for certain kinds of problems, like understanding motion or gravity. They weren't designed for understanding life. If Newton had started from the problem of trying to understand biology and life, I think the structure of his equations would have been quite different. And even sort of the mathematical formalization. And so the main point I just wanted to make here is that I think we really need to think outside of the box of how we've been trained as physicists to think about how the world works in order to think about what biology is doing. And I really do think it represents a new physics. And when we get at that new physics, we can worry about how it's unified with the other physics that we have, in the same way that we've worried about unification of different domains of physics in the past. We have a long history of that. In fact, in Frank's paper, he has a whole list of all the unifications that have advanced human understanding. They include things like unifying computation and or unifying logical thought and computation in the 1900s, which I think needs to be brought into physics in order to accommodate life. So that was, Turing and Boole and all of this kind of stuff that developed with theories of computation in the 1900s. So this is all just to say that I'm not sure that we're asking the right questions when we're thinking about what life is. And we might not even be seeing what's really there. So certainly, like when I'm talking to you guys, I'm thinking about all of the humans on Zoom being life, but I'm not thinking about my computer, for example, as being representative of life or living physics, but maybe it should be included in that category. And that's just my subjective bias. So that's one example. But I wanted to kind of give it a little short example from the history of physics that I think is kind of deeply telling about sort of what we might be missing about descriptions of reality when we're thinking about what life is. And so one of them is sort of just to think about the sort of evolution of our understanding of planetary motions. So thousands of years ago, sort of state of the art in building predictive frameworks for planetary motion was to develop models that we call epicycles. So Ptolemy obviously had this really nice model of planetary motion, which had circles inside of circles. And part of the motivation for having circles was they were perfect objects. And of course, the heavens should be described by perfect objects. And so they were really quite predictive at describing things like the retrograde motion of Mars. But we realize now that that obviously was a good descriptive model, and it was even predictive, but it wasn't explanatory. And it took, 1,500 years roughly before Galileo and Newton, came to the realization that the motions in the heavens and the motions on Earth were based on the same principles. So that was actually a major unification in human thought, right? Terrestrial and celestial motions being described by the same thing. So Isaac Asimov, captures this in that we all know things fall, but Newton was the first person to discover that the moon falls too by the same rule that we do. So you can think about, like, most of human history, people didn't know that that was the case, but Newton was able to figure that out and develop mathematical theory to describe that. And certainly we can describe planetary motions by Newton's theory, but Einstein came along and he was describing very different physical system. He was very interested in the properties of light, but that led him to the conclusion through logical deduction and reasoning that space-time is curved, right? So if I was sitting in this chair 2,000 years ago, I mean, it wouldn't be this chair, it would be another chair. There's no direct perception I would have about the curvature of space-time around me, and that's why I'm actually stuck to my chair. But it's there, and we can figure those things out, and they become more explanatory theories for how the world works. And I think when we're talking about life, we're still in the epicycle phase. We're in this kind of descriptive, maybe we can make some predictives theories, but we haven't gotten to that explanatory framework from which all of those other properties that we see should be derived. And I think the sort of main dichotomy about why we haven't been able to do that yet is really nicely captured by this quote from David Deutsch in his book, The Beginning of Infinity, where he says base metals can be transmuted into gold by stars and by intelligent beings who understand the processes that power stars and by nothing else in the universe. So what he's really talking about is we have physics as we've described it, which tells us about the laws in the world and how they operate. And then there's this other physics that describes that there's these things that we call intelligent or living things that somehow understand those rules and they can actually control those processes. And that's a very different kind of phenomena. If you wanted to think about it in sort of fundamental physics, how we talk about it, you can think about it as physics of the observer. But I think when you think about it from the biology, life, origins of life perspective, you get a slightly different perspective on it than that. And so this kind of brings me to sort of the way I pose the question, which is to ask, what are the things in the universe that we can't explain right now, or would be really hard to explain or account for based on our current laws of physics? And these are just sort of two very anthropocentric examples, but I think they're supposed to highlight the point. So one of them, the one I've shown on the right here, is the periodic table of the elements. And I think this is kind of very telling because you can go through each element in the periodic table and you can talk about the physical processes that can create that element in the universe in high abundance and do so reliably in the sense that you would expect that process again and again to reproduce those elements. The interesting thing is if you get to the highest atomic number elements like Einsteinium and fermium, and Mendelevium, and then all the way up to 118, which I think is like what the highest atomic number element. I can never remember the name. It starts with an O. But anyway, those elements can only be reliably produced by technology. So what that requires is that you have a physical system that has acquired some knowledge of nuclear physics and can constrain and control the conditions to such a degree that they can actually produce that element, and they can produce that element again if they, so like if that physical system was set up to be repeated. And so there's this idea that somehow the process of making those elements in high abundance in the universe requires some information about how to do that process. And the ability to actually set up the constraints for those conditions to occur. So those elements are really, as far as we know, the only physical processes that can produce them reliably are technology. The same thing with thinking about our planet and the fact that we're the only planet that we know right now that's anti-accreting. what that means is, so like planets form by accretion, right? So we clump matter together and eventually we make planetary bodies. David Grinspoon has this nice concept of anti-accretion as being a process that happens in the Anthropocene, but it's also just something that you might expect a globally organized technological civilization to do. What are we doing as a physical phenomena? Well, we're launching metal boxes into space. We're doing it repeatedly, but that process requires knowledge of the laws of gravitation. And so there again, you require a long evolutionary history of acquisition of information and control of certain physical properties. And if you think about what the laws of gravitation are, they're the ability of a physical system to extract regularities of the physical world, and then to be able to perform transformations that they wouldn't do unless they actually understood those regularities. So part of the argument is anti-accretion doesn't happen on planets in the way it's happening on Earth unless you have an evolutionary process that's discovered the laws of gravitation. So a colleague of mine, Michael Lockman, and I have thought a lot about this kind of idea of like, well, what do we mean by information and how do we think about this in the debate about definitions for life? And one of the things that we try to propose, at least as starting to conceptually clarify some of the issues, is to make a distinction between what we mean by life and what we mean by alive. And I don't necessarily like using these words for these things, but I think it's provocative enough to at least jolt us into thinking differently, which was kind of the goal. But I don't know if if we come up with final theory, I would actually call these things that. But the idea is that life is sort of the set of objects that could exist in the universe, but require information to produce them. So that would be things like cups, me, my computer, Zoom. And so all of those things are life. The argument being that they would not form in the universe unless you had some information to specify their formation. Information being a very vague word here, and I don't mean it in the Shannon sense, I mean it somehow in a causal sense, but this is part of the theory to be constructed. Some people might argue that cups could spontaneously fluctuate out of a quantum vacuum, or Boltzmann brains could, and I think that's actually the wrong physics. I think they require an evolutionary history of things that exist acquiring information about how to make more things exist, like objects. And so you don't get these kind of objects in the universe unless you have that history. And then alive things are a little bit different. They're the ones that are actually actively processing information and sort of traversing the space of what's possible and constructing things. So some of these ideas are really deeply related to what John von Neumann was talking about with universal construction and things. But the way that we talk about it and the sort of formulation I'm working on with Michael and Lee Cronin at Glasgow and Chris Kempes at Santa Fe is called assembly theory, which I'll talk about at the last arc of the talk if I have some time. But just to kind of make this point, so when I think about what life is, I think of life as a kind of physics we don't understand that emerges in chemistry and has to do with the combinatorial space of chemistry and how very complex systems can traverse that combinatorial space and make more complex objects. And they do that somehow by processing information or acquiring knowledge. And so I think about life sort of as a continuum where some systems are more alive than others, and there's a transition from non-biological systems to biological systems where information starts to actually matter to matter. In terms of actually mattering to dynamical outcomes. And so sort of the first principles approach I have to trying to derive a theory for life is to say that life is the physics of how information structures matter. And when we talk about an example of life, that's an origin, a life transition, and then that process propagating out in space and time in what we call the evolution of a biosphere. And so this is kind of just to like go back to the gravitational physics analogy. So my view is that life is the phenomena where information is the dominant physics. And so just to give sort of a framing of what I mean by that, we're pretty well familiar with the idea that like the laws of gravitation are universal. And so they should apply on Earth and they should apply on exoplanets, et cetera. We need some universal principles to describe life if we're going to be able to look for it in the universe. So hopefully those kind of physics are there. But the argument here would be just like you go to study a massive body or a black hole to study gravitational physics, because that's where that physics is dominant, or at like galactic scales, but gravity doesn't really matter much inside the nucleus. Information is a universal physics, but it only really matters when you get to a certain scale of chemical diversity and combinatorial diversity. That information actually has to be there for those objects to exist. And so it's sort of a probabilistic argument, but the idea that we have in mind is that the original life is an event that crosses some threshold in this space, combinatorial space of things that could possibly exist and actually allows certain structures to exist. So things like a protein you won't expect to spontaneously form in a prebiotic environment. They require an information system that can actually construct a protein to form. But a simple molecule like methane can form. And so the origin of life transition is somehow when you cross that divide and you get these information processing systems that can actually do that. And so we're looking for when we're trying to answer the origin of life question.
Yeah, let's touch base by email. Sorry, I have to go. There's another conference.
Okay, I'm almost done with this part of my talk too. So we'll pause for a break because I think I want to make sure that we have time to talk. But so from my perspective, what we're looking for when we're looking for the origin of life is not just a transition in math, like in chemistry, or the emergence of a replicating system or something. I think those are artifacts of the physics that we're interested in. But it's really a transition from physics where information doesn't really matter much to physics where it's the thing that matters most. And so I obviously, like, we don't know what this theory looks like yet, but I think trying to conceptually reframe the problem is important. And some of the things that come out of this set of ideas, I think will ultimately end up being important to whatever theory it is that describes life. And a lot of those have to do with the fact that, like, we're so focused on like a cell as a fundamental unit of life. But what this framework suggests is it's actually the lineage and the propagation of information through time. That's the way you should think about fundamental units in biology. And some of those things actually are very characteristic of life across the board, independent of whether you think the kind of framing I'm offering is the right one or not. I just want to say, like, I'm going to talk mostly for the rest of the talk about just a couple areas of research that my group does, but to try to get at this physics, we're thinking about it from a lot of perspectives, because it's like when you think about the life problem at this sort of broad and abstract scale, you have to bring a lot of ideas together. And so what I'm going to talk about for sort of the second arc of my talk is how we can look for statistical patterns in biochemistry and kind of apply ideas from statistical physics into thinking about the organization of life in chemical systems. But we also do a lot of stuff with information theory, which I would love to talk about because I know a lot of people here are interested about it, but I had to like kind of choose what I talked about today. But like how informational patterns and contribute to like collective behaviors in biological systems. And we work in systems from ants to plants to xenobots, which are programmed living materials. I will talk a little bit at the end about this assembly theory stuff, which is kind of related to the network stuff and how we're thinking about information and chemical systems. And then we do a lot on exoplanets and thinking about looking for life and also consciousness and nature of intelligence. So I'm going to stop here for a minute in case there's questions, and then I'm going to move to sort of the next part of my talk and check how I'm doing for time. Okay. Are there any questions?
I have a quick question.
May I ask a question?
I don't know who is first, so, and I don't see neat, like they're not coming up. Oh.
So I put in the chat 2 questions.
Okay, I don't have
This is Hiralal John. So the one thing, I mean, actually at the practical level, we already have like, physics and chemistry coming in protein folding, for example. Before that, what Schrodinger said was DNA structure we determined. Now we have, after the genome sequencing, we have the complex biomolecular networks, and then we have the complexity theory and information theory and entropy. So we do have sort of outline of what should happen, right? I mean, we can measure it, what should happen. I mean, it's true that you have all theoretical physics out there and chemistry out there, but we sort of have a foundation for what should be, right?
You mean in terms of the structure of proteins? I'm not sure what you're.
No, I mean that we already have how chemistry and physics comes in play in
Oh, sure.
life, in
Right, but I think there's
so
plenty of examples.
what
Yeah.
we can predict.
Yeah. So
And the second part was, I mean, are you going to quantify in terms of like information theory, entropy, complexity?
No, I don't. I mean, I think there's elements in those theories that are useful, but I think ultimately they're not exactly framed properly for the problem. Here's a sort of simple example of like I think the argument you're making is we have good physical and chemical descriptions of proteins and how they behave. And so is the argument there that there are examples where we can apply physics as we know it to biology and explain things. Is that sort of the, or so there's always this kind of thing I find really interesting, which is there's some areas where people want to say physics as we know it is explanatory biology. That typically happens in like non-equilibrium thermodynamics, because we can say life is a non-equilibrium system, and clearly that's the case. But the Great Red Spot of Jupiter is also a non-equilibrium system that sustained itself for hundreds of years. So I don't think it's explanatory enough. And then on the other side of it, there's you'll get kind of odd communities that want to explain life in terms of like quantum biology, for example, where there it's sort of the argument is, well, life uses some quantum effects, so therefore it must be quantum mechanical in nature. But I think the problem is life cannot violate the laws of physics, right? It lives in a physical universe. It has to actually be able to use those laws for the functions that it needs to perform. And so nobody talks about like the fact that life does gravitaxis, is trying to use gravity to explain life. Life uses chemotaxis. I mean, it's a general characteristic of life that selection has learned to operate within the constraints of the laws of physics. It couldn't do otherwise. So I don't really buy that as an explanation for what life is or what life is doing. I think it helps constrain what are the physical media and the properties of those media that life can live in. Or exist in, but I don't think it explains what life is. Does that answer your question?
No, then it becomes more metaphysical. But anyway, thank you very much.
No, it is. I mean, it is getting metaphysical, because I think you have to kind of go to the metaphysical territory to ask what's wrong about the way we're thinking about it now to come up with an explanatory framework. And the reason, I mean, I think when you study life and you're not worried about the origin of life problem, you don't have to think about it at the level that I'm requesting, because there it's very clear that there's a consistency between physics and biology, but you're not trying to explain the origin of biology. And there you're confronted with really different sets of questions. And I think it's really important to recognize that.
Thank you.
Sure.
I had a question too, Stephen
Yeah.
Watson. And it actually links to the remark that was made there. So speaking of thermodynamics or concepts of entropy, those do connect to what physicists would call equilibrium thermodynamics. So if you look at active matter systems, those are a place where indeed we're still struggling to formulate universal thermodynamic principles for non-equilibrium systems. So I guess, are you viewing those types of studies as stepping stones also in your effort to identify universal features?
Yes. Yes.
Yeah, super. So, and so when you talk about the physics of information, am I to interpret that's in some sense linked to this notion of universal principles, which some physicists might call thermodynamics for large scale systems, where you're trying to capture what it means to be out of equilibrium or driven?
Yes. Yes. I think I agree with all of those things. And I should say, it's not that so obviously, it's not like you want to throw away the theories we have. You just want to ask, like, where are they not explaining things? And we need to, come up with new ideas. Oh, sorry, go ahead.
No, so I would focus on the notion that indeed what's missing there is capturing the emergent physics. So in some sense, the physics is not yet known. The large scale collective behavior of lots of driven active matter systems we don't have. So in some sense, we're still trying to find the physics.
Yes, I agree with that very much. And I think one of my main points is we don't, if you look at what life is doing across all different scales and you're trying to build an explanatory framework for that, we don't know what that physics is. That's all. And when I use information, I actually mean something quite different than most people think by that concept, because I mean something closer to what we would think of as causation. But there's no notion of causation in physics. So there's kind of a deeper set of issues associated with that.
Yeah, so maybe I just feed one last thing in. So, sort of self-organizing systems which are pattern forming, which are in this realm of active. I think those are interesting to think about from your standpoint, because in some sense, not only are you trying to understand the emergent patterns, these, if you like, statistics of shapes, but in some sense, the language to encode it and how much information is there is in some sense completely unknown.
Right, right. And so part of it, and I guess from the language that you're talking about, part of it is what patterns emerge also dictate future dynamics. So it's not like you can decut. So this, one of the issues is you can't really decouple the way the rules behave from the way the system is at that given point.
Yeah,
in time.
but there's one feature which is a simplifying feature for many of these systems, which is dynamic scaling. So
Yeah.
under certain sort of scaling transformations, there's something universal and unchanging.
Right. Yes.
But
Which is a good
parameter.
segue into the next part of my talk, because I'm going to talk about universal scaling in biochemistry. But...
Okay, super. I'll let you go. I'll let you go on. Thank you.
Yeah, no, it's good. I mean, there's so many things to unpack and talk about here, and unfortunately, there's not a lot of time to go into like the semantics of, you know, the different ways we talk about things. But it's good.
Sarah, let me also mention that there are a bunch of questions in the chat as well.
I know, I see that coming. Should I answer more questions now, or should I proceed? I'm not sure. I'm happy to follow whatever you guys would prefer.
Up to you. I think you'll probably get more questions than there's time to discuss. So I'll let you decide which question to go.
Okay, I'm going to move on to the next part of the talk. So, so a lot of the ways that I've been, you know, kind of thinking about some of the principles that might explain things are to think about sort of concepts from the way we think of physics that might port over to answering some questions that we have in origins of life or astrobiology. So the one I want to focus on today is just thinking about this notion of universality. And also thinking about the sort of nature of the problem when we're talking about looking for life in the universe. So, so I already mentioned this, but you know, when we're talking about astrobiology and the search for life in the universe, we're making an implicit assumption that life has universal features. If it didn't have universal features, there wouldn't be a category called life, and there would be no point to looking for it. So one of the questions we have in astrobiology is sort of how much of the physical universe organizes into things like us, whatever that means. And I just put it on here because I'm trained in cosmology, and I think it's fun to think about the fact that we feel like we know so much of the universe, but we don't really understand this really important part of it. Universality in biology and biochemistry is kind of one of the issues with trying to build general principles, is that when we talk about the universal nature of life on Earth, it's not translatable to the universal nature of life everywhere, because we're constrained by a common history for life on Earth. So if you try to trace the lineages of all known life back in time, what people that do that kind of work have done is basically be able to construct something that's called the last universal common ancestor of all life on Earth. It probably isn't a single cell. It was probably a community or population of organisms or entities that lived on the Earth sometime between 4 and 3.5 billion years ago. And it has all of the sort of key components of the cellular machinery of modern life. So it had DNA, RNA, proteins, and unclear, I think still of what its membrane was. But I think there's some people on this call that might know more about that than I do. But in any case, so this is very constraining, because it suggests that at least on Earth, we have evidence of only one origin of life event, because everything that has come subsequent can only be traced to this last universal common ancestor. And in some ways, you can almost think about it just using our physics analogies, is at the genomic level that LUCA is like a surface of last scattering. So it's really hard to get information about what happened before LUCA, because you can't use genomic reconstruction to go to that scale. And so when we're talking about origins of life, there's actually this fundamental gap, because we have the prebiotic chemists that are trying to synthesize components of life, like amino acids and nucleobases from prebiotic conditions, which I could talk about sort of some of the things I like or don't like about that approach. But then there's also, you know, tracing back to this last universal common ancestor, which is a fairly complex organism or population of organisms. And then there's this big gap in between. And so a little bit like the first 3 minutes of the universe, but as we have to actually be able to try to trace past that. But the signature of that sort of that last universal common ancestor event is distributed over the entire planet now. So we do have kind of a signal in the genomes of extant organisms, and that's basically how we've been able to reconstruct the last universal common ancestor. So one of the questions that we could ask is, what other information could we extract from the distribution and properties of life across the modern planet? And so that's sort of one of the questions that's motivating a large fraction of my research group right now, and trying to think about statistical patterns in this chemistry that might allow us to extrapolate back past Luca and understand properties of the earliest chemistries for life. And so this is sort of questioning the nature of what we talk about as universality in biochemistry and trying to come up with new notions of universality. Part of the thing that I mentioned is that when we talk about universality across life on Earth, we're talking about this shared biochemical architecture. So what we mean in biochemistry by the concept of universal biochemistry is that all life on Earth shares some of its chemical components. So some people have argued, in particular Norman Pace, that the basic building blocks of life anywhere will be similar to our own in their generality, if not in their detail. The problem is that life, at least for astrobiologists, when we try to generalize beyond life on Earth, right now we're focused on the detail because we don't have any better principles for doing it. So for example, like the Mars rover, Perseverance just landed yesterday. A target for looking for things might be to find biological amino acids. But we wouldn't know what patterns to look for in chemistries beyond the specific chemical details of life as we know it. So how do we go to this better notions of generality? Well, I'm obviously inspired by thinking like a physicist, but we do have a different concept of universality in physics. It's very different from the universality in biochemistry. So universality in biochemistry is about universal component membership. So every example of a system you see has some of the same components. Universality in physics is there's some macro scale properties of the system, not details, that display universal behavior. And we call that kind of phenomena universality class. And we're really familiar with that, for example, in the physics of phase transitions, where we might see very different physical systems display similar behavior in a scaling limit. And so this concept of universality seems much more powerful because it allows you to describe properties of different systems that differ in their specific details, but talk about the universal physics that might apply to them. And so one question that we've been asking is whether there's any notion of universality in a more physical sense that we could apply to biochemistry. And to do that, we need to construct ensembles, because if you're talking statistically about systems, you actually have to have ensembles to look for statistical patterns. And so most of our data comes from the Joint Genome Institute, and we also use annotated functions for enzymes that are in the Kyoto Encyclopedia of Genes and Genomes. And basically what my group has been doing over the last several years is constructing an ensemble of thousands of data points of curated genomes or metagenomes. So metagenomes are environmental samples. So if you just go into a hot spring and you take a sample and you don't care about the identity of the individuals and you do a sequence on the genomes that you get, that's what a metagenome is. So you can think of it as a community level genome. And then the biosphere level that we look at is just to look at all the cataloged enzymatically catalyzed reactions. So you can think about this as studying the reaction space that biochemistry lives in. We're not talking about it as a dynamic space. We're just talking about the structure of the molecules and the relationships between them through reactions that biochemistry uses. And then we're trying to look at it across scales, looking at individual species or taxa, and then ecosystems, and then the biosphere as a whole. And so this is sort of something that we just call planetary systems biochemistry. And so when we're thinking about life on Earth, we're trying to abstract it to thinking about the chemical components of life on Earth and their relationships. So this network that I've shown here is just a network representation of the biosphere as a whole, as far as all of these cataloged reactions that biochemistry does. Nodes represent compounds, so these are molecules that participate in reactions, and they're connected by an edge if they participate in the same reaction. And the size is indicative of how connected they are in the graph. So for example, these large ones are likely ATP and water because they participate in a lot of reactions. So things that are heavily important in biochemistry obviously would play a larger structural role in the network. And what we're looking for is scaling laws that apply universally across biochemistry, but aren't necessarily observed in random chemistry. And so we can look at different network properties or statistics of different kinds of reactions or properties of the molecules themselves, and then do that as a function of system size. So I've written network size here would just be number of molecules, but there's a lot of different ways of doing this depending on which scaling laws you're looking at. So I'm going to show two examples of scaling behavior that we found in biochemistry. The first one was published in Science Advances 2 years ago now, where we demonstrated that if you look at this ensemble of thousands of individual taxa across bacteria, eukarya, and archaea, and then you look at thousands of community level biochemical networks, they have universal scaling behavior as far as their topology, but also in terms of the reaction diversity and some other properties that have nothing to do with the network projection. So it's not projection specific if you know sort of details to think about networks. But if you look at number of compounds and then you look at average source path length and clustering coefficient, just as two examples, there's more in the paper, we see we can write scaling laws and scaling behavior for them. And then the biosphere is consistent with most of the scaling behavior, but not all. And when we're thinking about this ensemble, it's important to recognize that the features of the last universal common ancestor or the shared ancestry show up very prominently if you think about the composition of individual organisms. So each living system on planet Earth is a partition of this global biochemical space. So basically an organism has a subset of the reactions, but there's a lot of overlap in the reactions that they share because of this nature of universal biochemistry and the shared ancestry of life on Earth. So one of the things that we wanted to ask was, are these scaling laws a generic feature of chemical systems, or is it something specific to biochemistry that we might say becomes a law of biology because there's some kind of evolutionary optimization or some other principles that are operating in biochemistry that aren't present in chemistry? And it turns out this question is really hard to ask, because if you want to ask for random chemical networks or control chemical networks, abiotic systems, like that data is actually really hard to generate and get, even if you have access to the world's largest chemical databases, which we happen to be lucky enough to have. So what we did for this paper was actually to randomly sample biochemical space, but do it in a way where we weren't conserving that universal core. So we just did sampled reactions, ran completely randomly, but not biased in any way by their frequency distribution across life on Earth. As a first set of control. And so therefore, what we see is that the universal scaling behavior is not conserved. There's still a scaling behavior, but it doesn't have the same exact behavior as what we see for real biochemical systems, which is suggestive that the real biochemistry has obviously additional constraints or properties associated with it. And in fact, I would expect that if we just randomly sampled things that weren't from biochemical space, they would differ even more. So this is just demonstrating that random sampling chemical space doesn't recover the scaling behavior that we would expect for biochemistry. If we do a frequency-based sampling, so we're conserving that universal core, we get much closer to the real scaling laws that we observe in biochemistry, but they're still statistically distinguishable. So there's something, so this random sampling is not conserving, say, all the functional components of real organisms or other constraints that we could have set, but it does recover most of the features. And we also did some randomly sampling where we did sampling genomes to try to recover the scaling laws for real ecosystems or community level genomes. And there you do recover most of the features, but they're statistically distinguishable as well. So all of the random ensembles that we generated scaling laws for were still statistically distinguishable from real biochemistry, but the frequency-based sampling and the genome-based sampling, which recover features of this kind of universal core ancestry, recovered much more of the scaling features that we observed. And so our kind of conclusion is that a lot of these features of the scale invariant topology of biochemical networks requires this universal set of biochemical reactions across life on Earth. And so the reason that we see the particular scaling features are because of that common sense set. So it could be just sort of an ad hoc artifact of the early evolution of life on Earth. But I think there's also another way to read it, which is that maybe part of the reason that we have a last universal common ancestor is because there's constraints on the global organization of biochemistry. So if you think about the fact that all life on Earth is connected through this space of chemical reactions, there are some global constraints on that reaction space. And when you have the emergence of individuals in early life, it might be that those constraints actually make it such that organisms actually have to share a subset of reactions. So it's a little bit of a different narrative than this sort of evolutionary radiation, and it's not inconsistent with it, but it's another set of mechanisms that we might explore for why the last universal common ancestor has this shared set of reactions. It comes from this idea of these global constraints and scale invariance of biochemistry. So my hypothesis is the last universal common ancestor is in fact an artifact of the fact that biochemistry itself has to be scale invariant and display common scaling features across individuals, ecosystems, and all the way up to the biosphere as a whole. So I'm going to
could I ask a quick question?
Absolutely. Yeah.
So is that scale invariance something that you understand the actual exponents for, or that's just that there is?
No, we don't have a theory to derive the scaling exponents, and I'm not actively working on that for the network properties right now because for the reason of the work I'm going to show next, which I think is a little bit more telling about the actual physical mechanisms, because what I'll show next is that we also see scaling behavior in the actual reactions that biochemistry uses. And I think there's a much easier route to a mechanistic understanding and connection to more fundamental physical principles than we would get with the networks. But it certainly would be possible, I think, to construct, some simple network models that might recover our scaling coefficients.
So just to throw in some language from my perspective, it's interesting if those exponents are emergent, or what physicists call anomalous, versus intrinsic. So you can have scaling symmetries that are present at the microscopic level, but a scaling exponent which isn't consistent with those.
Oh,
so in that sense,
talk about that. That would be really great.
Super.
Yeah, I would love to talk about that. The other thing I can say is I did some work with my group, which we haven't published yet, but I'm trying to write just a concept paper on this idea right now because I wasn't sure where to go with it. Trying to demonstrate that this scale invariance required something like a set of shared reactions, so some universality and component membership, a last universal ancestor type structure. And so what we did to do that was actually vary the overlap between networks, like with some random models, and also and then what we can show is actually what you see is a series of phase transitions in the structure of the network. So all of the networks in biochemistry are roughly scale free. They're not exactly scale free. And then they show this common structure across scales. But if you vary the amount of overlap, so they have almost no overlap or just a little overlap in their set of reactions, you start to see networks that are much more random in their topology emerging as you get to larger and larger scales. And so it might be the case that you could have individuals that are scale free, but the global network of biochemistry would not have a heterogeneous topology, but it'd be much more homogeneous. And so this was very suggestive to me about this idea that you have to have these constraints operating across scales and this heterogeneity in the networks across scales, which I think gets into some of the stuff that you're talking about. So we have a lot of data on that. Would be super exciting to develop theory. Yeah, so I just want to kind of get into the next part on the scaling bit, which is this idea that universality is usually related to coarse graining in physics. So obviously we're familiar with the idea that temperature is kind of an effective description that averages over the properties of molecules in a gas. I don't think I need to explain coarse graining for this crowd, but the nice thing for us anyway was that we really were interested in sort of understanding some of the reactions that biochemistry uses. And it turns out that the way that they're actually cataloged is an effective coarse graining of chemical reaction space. So each enzyme has a function. Actually, enzymes can have multiple functions, but enzyme functions are the particular reactions that they catalyze, and they're cataloged by this 4-digit identifier that's grouped hierarchically so that reactions that have a common function are grouped together. So you can kind of think of this as a hierarchy of coarse grainings, and at the coarsest scale, you sort of have the major reaction classes that we talk about in biochemistry, and there's actually 7 of them, but there were, when we were doing this work, there were only 6. So that were heavily annotated. The 7th is a newer class, but these reaction classes describe sort of the major functions of biochemistry. And so what we wanted to do was actually look at these as sort of a macro scale description of biochemistry where you've coarse grained by function, and then try to look whether there was any regular scaling behavior associated with that. And so this is sort of just the pipeline. We take the genomic data, we look at the different functions for the different enzymes, and then we group them and count how many of each kind of function there is in archaea, bacteria, eukaryotes, or metagenomes, and then we look for scaling behavior. And the results are that we see very regular scaling behavior for all 6 major enzyme functions. If you actually look at the scaling coefficients, they all are more consistent with a power law fit than other fits that we tried. Just that there are some enzyme functions that display universal behavior, whether they're superlinear scaling, linear scaling, or sublinear scaling. So for example, the oxyreductases, which are responsible for redox reactions, which are electron transfer, so associated with energy use in biology, scale superlinearly across all of our data sets. And so do hydrolases, which are when you break a bond by releasing hydrogen. And then ligases are reactions associated, sorry, I'll go back to the functions so you can, that with joining large molecules, and they're essentially really related to sort of core functions associated with DNA and RNA. And so the ligases actually are sublinear in their scaling universally, and so are the transferases. So there's only 2 classes that don't display behavior that's consistent across the data sets, but even the isomerases actually could be considered to be sublinear because their coefficients actually consistent with it. So the main sort of take home that we took from this is, at least in most of the major enzyme classes, biochemistry displays universal scaling behavior. There's variation between the data sets as far as the scaling coefficient, and there's really tight, error on our scaling fits. But it's suggestive of this idea that there might be sort of universal principles organizing biochemistry across these different major divisions at the level of individuals and across ecosystems that are universally shared. And so we think that this enzyme class scaling is actually a really good candidate for universality class in biochemistry. And one of the arguments that we wanted to make is that it's more like a universality class in physics than being explained by this idea of shared ancestry and a common universal ancestor for life on Earth. And so that would require that the scaling behavior that we see is not fully explained by shared component parts across life on Earth. So if you can sort of disentangle the fact that we see universal scaling behavior from the fact that there's shared components across those data sets, then that would suggest that that scaling behavior was more of an emergent property and one that might apply to other biochemistries and have some physical principles that we could use to describe it. So to get at that, what we did was try to take each enzyme function in each data set and rank order them by how common they are across that data set. So for example, this data point that I'm pointing to here in archaea would be an enzyme function that is present in every archaea in our data set. So what we're able to show here is that the ligases, which you might expect are more universally distributed. And part of the reason you might expect that is because they're associated with sort of core information processing machinery in biochemistry. And then the oxioreductases are less universally distributed. So they tend to be less frequently found for a specific function across organisms on Earth. And to quantify that, we used area under the curve scores. So we would assign a score which basically measure the area under each of these curves. And that would be sort of a score for the quote unquote universality of functions in that data set. And scores closer to 1 are indicating that function is more universally distributed across that data set. So ligases are very universal in metagenomes. So most ligases are found in most metagenomes. But if you look at something like oxioreductases, they're very rare in individual genomes or individual genomes sampled from the different taxa, but they're more common in metagenomes. So there's a general trend here where we see things are more universally distributed in metagenomes, and also that oxioreductases are less universally distributed than some of the other enzyme functions, and you can talk about sort of all the variation in them. But the key point that we were able to show is that there's not a correlation between the universality that we see in scaling behavior and the universality that we see in component membership. And a good example is the fact that the ligases have a lot of variation in their scaling coefficient. They vary by about 0.25, but they're the most universally distributed. And if you look at the ligases, for example, their variation is less. A lot less. They're pretty tightly constrained across all the different data sets, but they're less universally distributed. So some of the components that are most universally distributed have the more variation in the scaling behavior and vice versa. So we feel confident we can make the claim that these two things are decoupled. And so this brought us to starting to ask questions about the last universal common ancestor. So in this paper, which is now up on the bioRxiv, we have a lot of data about the universality of component compounds and different things associated with where LUCA is in modern organisms. But the LUCA model we used was actually generated by one of our collaborators, Aaron Goldman, who took the 8 leading models for the last universal common ancestor, which are constructed by different methods. And he took sort of the functions that were common in them and constructed a consensus LUCA model, which is the one that we're using for this study. And the main point I want to show is that if we look at the scaling behavior on the smaller size scale that the last universal common ancestor is on, the last universal common ancestor is consistent with the universality class of modern life for some functions, but not all. I'll just point out in particular the ligases, which weren't too surprising that they are, quote, they're overrepresented in LUCA with respect to what we would predict based on our scaling laws that we've uncovered, probably in part because ligases are universally shared across life on Earth more than other enzyme classes, and therefore they're more likely to be present in models of the last universal common ancestor. So this allows us to start asking questions, are we overrepresenting or underrepresenting certain enzyme classes in LUCA models because of universality of component part, and could we build more constraints based on the scaling behavior and the predictions we might make about early life and even simpler systems beyond LUCA if we can explain why we're seeing the scaling behavior that we are. And so sort of just as a little sidelight, because I know people here are interested in this, and I'm really happy to talk about it more, but this is just some work that my group is really starting to get into, is to think about the fact that we've done the coarse graining at the highest scale of reaction diversity in biochemistry and shown there's regular scaling behavior and potential for universality classes. But there are these subclass functions. Remember I said that the enzyme classification is actually a hierarchy of coarse grainings. So it has some of the kind of feel of what we talk about, say, for example, in the renormalization group in physics, where you can look at coarse graining across scales and be able to come up with some regular behavior and explanatory frameworks for why you see the particular structure that you do. And so I'm interested in whether this renormalization approach applies to biochemistry. In particular, it would be kind of an interesting case because you're grouping by reaction function, not any kind of spatial variation. And just to offer some suggestion that there actually might be something here. EC1 was the oxioreductases, and these are just some of the subclasses of oxioreductase shown here, generated by one of my students, Veronica Merzinski. And we see really regular scaling behavior for subclasses of the enzyme commission numbers. And there's a lot of really interesting variation in the scaling behavior depending on the subclass and what its major class is. And not all of them are totally consistent. So I think there's really interesting theory to develop here. And we're trying to do some theory work inspired by renormalization group and maybe trying to make some connections to like information theoretic approaches. And I think there's some really interesting insights about thinking about life as a planetary scale phenomena, because the information about planetary context is probably encoded in this coarse grain hierarchy. So my long-term vision with this is to build better models of biochemistry on exoplanets, but that's sort of a long-term goal. But what it does afford us is this possibility, like I was saying before, of reconceptualizing the last universal common ancestor as a planetary scale phase transition. So a lot of people have talked about the last universal common ancestor as a phase transition, but I think the thing that we're trying to bring to it, sort of that's very heavy data driven and it has a lot of potential for developing theory based on the scaling exponents and features we're seeing, is to actually say that this is a planetary scale phase transition mediated by the fact that when you get a living planet, you have to have the scale invariance to the biochemical architecture. So that's sort of the hypothesis that we have there. So I'm going to stop for a minute. I have one final part of my talk if we have time, but I'm also happy just to stop at this point for questions too, depending on what people want to do.
Do you expect some sort of universal like preferential attachment laws for the networks? For example, because you know, is there a universal law like preferential attachment? I mean,
oh, sure. Yeah, I mean, I think you could talk about the generation mechanism for the networks in terms of preferential attachment. People, I mean, we're working with the same network data that, for example, you know, Barabassi's early work on the large scale organization of metabolism, and then they showed, you know, the preferential attachment model for those kind of networks. So I think, yes, you can do that. I think the problem there is that actually biochemically realistic? Because there's a lot of different hypotheses about how pathways evolved, and they don't necessarily conform to what a network theorist thinks as far as network formation mechanisms. And there needs to be some bridge between those things.
Well, in your networks, it's not clear, you know, how you connect them. What is the strength? What is the signal strength? I mean, they're weighted networks, but you don't.
They're not weighted. So we have a separate paper, which I don't talk about here, where we did, so the projection matters. And then also like if you weight the network and if you have directionality on the edges and these kind of things. And this is one of the reasons that I wanted to move more to this reaction mechanism framework, because I think there's more promise for developing physical theory and you don't have to worry about the issues of the network projection and if you're capturing the relevant features. But just as sort of one example, we did do this paper, which is supposed to be out in Science Reports soon, on looking at all possible simple projections of biochemical networks using some of Aaron Clossett's work on sort of, trying to statistically constrain if a system was a power law distributed or not. And just we're able to show that biochemical networks are not scale free and in general. But I don't, I think for personal reasons that I was just articulating, I don't think going down that road of worrying too much about the network structure is exactly the right way of thinking about it right now. And I'm happy to have a longer conversation about why, but I don't think it's going to get into sort of physical mechanisms of how these systems are organizing in quite the right way, unless you're asking very specific questions, like I was saying about universality of component membership and how that affects the topology. Like there's certain questions and hypotheses you can ask in the network representation and certain ones you can't.
Yeah, then you can apply information theoretic methods.
Yes. Yeah. That would be cool. Yeah.
Sarah, could I ask you to go back to the last plot that you had?
The Luca one? Was that the? This one.
Let's see. No, I think it's that one. Yes.
Yeah.
So, no, actually, yeah, there we go. Could you, so I get the idea that you're coarse graining essentially by the significant figures in the enzyme, in the classification scheme. But I'm not exactly sure I understand what's now on the axes.
Oh, sorry.
So the y-axis is how many things? Well, I'll let you.
Yeah, so actually, this is a very old plot, and I should have probably replaced it with a newer one, but my student's been doing DFL statistics. She's got like a massive data set of all the scaling behavior across all these different enzyme classes, and we're trying to figure out how to organize the data. So this is more just sort of like, wouldn't it be cool if we could do this? And we have some preliminary stuff that suggests it's possible. This is number of compounds on this axis. The other scaling laws I showed for enzymes, so this is a good catch. We're in terms of total enzyme diversity in that biochemical system. There is a transformation between the two, but there's an additional scaling there associated with that transformation. So
these are the number of compound, the number of.
So you can think of this as the size of the system, roughly.
These are the number of compounds that participate in the reactions that are catalyzed by these enzymes. Okay.
Yeah, and there's a trend. It scales with number of reactions. So you could, it's number of compounds in each biochemical system. But you can think about it like we think about it in terms of the size of the system. And then here is the number of enzyme functions in each class. So for EC1, this is the scaling we see for all oxioreductases. And then for a subclass, so I know the colors are terrible here, but each of these represent a subclass. So this is grouping all of these together and counting them together, plus a bunch of other classes that I haven't included here that are subclasses. So it would be like the sum of the scaling of all of these behaviors. But if I just look at this partition of that, of the system in terms of these functions, this is how this scales. And then this would be a different scaling behavior for a different function. And then this would be the scaling behavior if I group all of those functions together.
And since these are linear plots, it's a little hard for me to tell. Are these actually, so is the curvature at small numbers, at small values on a power law, or because they look kind of linear at large values?
Some of them are linear and some of them are super linear. So the oxioreductases are super linear and as a whole class, and most of the subclasses of oxioreductases are super linear. But this is where it gets a little weird to think about and where I would love to have some more conversations, because I just don't have the intuition about how to think about this yet. Sometimes what we'll see is like if you have a class and then you look at the subclasses, not all the subclasses have the same scaling behavior as the class. So some of them, like it might be that oxioreductases as a class are super linear, but some of the subclasses of oxioreductases might be linear, even sublinear in their scaling behavior.
Thanks.
Yeah, there's a lot to make sense of here, but I think, I don't know, I'm excited about thinking about it more and trying to develop theory, because I think it'd be really cool to be able to predict, coarse features of biochemical systems in this way. But
could I ask a question about other sort of forms of universality? So focusing on power laws is trying to capture scale invariant features. But you also have other possible universal features. For example, in a power law, you have a prefactor. Let's call that a rate coefficient.
Yeah.
And that itself could have universal features, but not captured by power laws. Right. So have you got an interest in going beyond
I do, definitely.
scaling law
Yes.
perspectives?
Yes.
Super.
Yes.
Yeah.
I have no preference for the formalization. I just want to understand what's happening in these systems.
So just a comment. So the renormalization group sort of philosophy that relates to, in physics, second order phase transitions, critical point behavior. But if you look at first order phase transitions, where you think of quenched systems, which are in some sense therefore active or driven, with external drive, there are formulations of scale invariance and beyond scale invariance theories that you can actually structure, which parallel the Wilson approach for critical points. So that might also be useful for you.
Yeah, that would, okay. I would love to talk about that more. This is great.
Super.
I'm very excited. Cool. Yeah.
Super. Thanks.
I think we'll have to follow up and have a longer conversation.
Yeah, will do. Will do.
Yeah, great. I'm going to answer the last question in the chat because I feel so bad that I've been ignoring the chat and just answering questions that have been, because there's been a lot of really great stuff in here. Let's see, there's some discussion. I think Hyman Hartman was talking about phase transitions in geochemical cycles. I'm not seeing, and I was trying to follow that. But the last question here is, when could the phase transition in prebiochemistry have possibly resulted in geochemistry transitioning to biochemistry or biology? I think that's kind of the question we want to answer. I guess I don't have a clean answer for that, but I'm hoping that if we can identify the universality class for biochemistry and understand the theory that predicts it, we can trace it back and try to understand why it's like, and we can do comparable things for geochemistry. So that's sort of the boundary condition that's harder because geochemical data is much more messy than biochemical data. But that would be kind of the hope, is that we would understand the features of geochemistry that mediate this kind of phase transition. And I think they're related to a lot of hypotheses that people have out there related to these things and have talked about. All right, shall I answer more questions? Are there burning questions? Okay. So the last section of my talk, I know I don't have that much time left. So I'll try to be really fast about it. It's just more kind of trying to, like, I talk very abstractly and philosophically in the beginning, and then more concretely with what we're doing on the data side, in like these sort of data-driven approaches to understanding organizational principles of biochemistry, to extrapolate back to origins of the second part. And then in the third part, I just kind of want to introduce how some of the like philosophical ideas and abstract ideas we're trying to construct in a theory that connects to origins of life experiment, and also might test some of the principles that we're trying to do in sort of the data-driven approach. So this is kind of trying to bring them together, but more specifically, just thinking about what's possible for origin of life like the field of origins of life has a lot of modeling and a lot of people doing theory in individual or experiments in individual labs testing specific hypotheses about origins of life, which are actually more testing, can you make certain molecules implicated in some hypotheses about the origins of life than testing the origin of life proper. So, sort of one of the soapboxes I like to stand on is, physics has done really well with intensive large-scale collaborations between theory and experiment. And I'm a little biased in that regard because I did a lot of work in particle physics when I was an undergrad and graduate student. And obviously there we are very familiar with having fundamental theory, driving experimental design, and massive, international collaborations. But I do think that kind of ideology and mindset is actually really important for origins of life moving forward, and eventually will be kind of necessary for solving it. So I just kind of want to talk about like how we're trying to develop that with some collaborations that I'm involved with nationally and internationally. And so I've made this sort of argument that we need new theory and that there's non-living physics and living physics. And how do we actually get at this connection? Is there any way to actually test these ideas is more important, because this was like a very abstract sort of representation of what I think are sort of the core ideas. And so this is actually work from a collaborators lab at University of Glasgow, Lee Cronin, who's one of the main experimental groups that we work with on origins of life stuff, in addition to Kate Adamala in Minnesota. And we have a project together trying to explore informational transitions in origins of life. where they've been trying to actually formulate a way of thinking about chemical space, kind of from this perspective of thinking about how much information does it take to make an object, or how much history do you need, or memory is actually encoded in the object itself. And so what they do there is, you can think of a molecule as a set of steps to produce it, and you can actually build a graph of different ways of making it. And if you take a molecule and you break it apart into all the component parts, you can build all the possible paths to make that molecule, and there will be a shortest path in that space. And they talk about that as the assembly index of that molecule, as sort of like the complexity to actually construct that molecule. And what you can really think about it as encoding is how much history is that molecule encoding about the kind of steps of formation of itself. It's sort of a proxy for that. But importantly, it's a complexity measure that they developed in the lab that can actually be measured, because you can measure it with a mass spec. And so because mass specs actually work this way, they basically work by fragmenting molecules. So there's a direct correlation between when you talk about assembly of a molecule and what you measure in a mass spec. And so this became, so this is just sort of assembly number of some molecules in the lab. And then this is the assembly number on this axis and the molecular weight. So what you'll see here is that high assembly, you have assembly number is not directly correlated with molecular weight, which a lot of other chemical complexity measures are. So that's really critically important. And that's one of the sort of the key features of it, is actually measuring sort of the structure of the molecule from a perspective about the complexity of building it, not necessarily things like molecular weight, which are just features of molecules that could be quite simple because they're repeating units. And so one of the things that Lee's lab has been doing, which my lab has been collaborating with them on, is actually trying to look at the distribution of the molecular assembly in molecules across biological, non-biological, and dead samples. So this is, I think, abiotic, dead, and biological samples. And then what they're able to show is that there's this kind of boundary around assembly index of 15, where you actually don't observe any molecules being produced except if you already have a living example. And so a lot of people are kind of shocked by this idea, especially in the astrobiology community, because they're like, well, surely geochemistry can make high complexity molecules. And sort of my retaliation to that is, well, geochemistry can't make a cup. So why would you expect geochemistry to make a molecule of arbitrarily high complexity that requires a certain number of orchestration of steps? And that boundary is very clear to see when you talk about something like a protein. It's not so clear to see in sort of the general space of possible chemistries. And what assembly theory is basically trying to do, at least in terms of this assembly map of the molecules of life, is talk about where that boundary is in terms of this general way of talking about a molecule, not as a molecule, but as an assembly space, where you quantify something about the complexity of building that assembly space, that object, from the number of steps, the shortest path to create it. And so this paper is in review right now, but it basically is some experimental validation that these ideas work in practice, at least for biosignature detection, and that you can use this as sort of a generic measure for measuring complexity of molecules related that will require information or a living process to build them. And so the idea is low assembly space molecules, low assembly number molecules, are part of the abiotic universe. And this gets the ideas philosophically I was talking about at the beginning, that the only systems that can cross this boundary and make these more what we might call complex, but we would call in assembly theory high assembly number objects, or very structured assembly space objects, is that it requires a living system, or a system that has information to actually make those things. And so in some ways, when I think about this from a physics perspective, I think there's some fundamental bound in the combinatorial space of possibilities that random physics and chemistry can't get past unless you have some information specific to the object you're trying to produce. And this should be some kind of fundamental property of the universe, getting back to sort of the ideas that we were talking about before. So working with Lee's lab, I get excited because I think some of the ideas that I was talking very abstractly and conceptually about are actually things that can be tested in the lab. Once you have this idea that mass spec is actually measuring some properties about how objects are assembled from component parts and what the sort of causal pathways might be for forming them. And so we can talk about this in terms of molecular space, and just in terms of like graph theory. You can build these graphs about sort of the assembly trees for molecules, and how likely it is to produce them under certain sets of constraints and things. And so there's been a lot of work done in that space. But for me, the more interesting thing is to think about this idea of assembly spaces. So this is an example of an assembly space just for a string. And you might ask, like, you know, how many ways are there of getting to this object? And then we talk about this whole subspace of that object as being its assembly space. And then we can talk about sort of the interaction of different assembly spaces and their dynamics as kind of the structure of the theory that we're developing. And so part of the idea of this is that we're going to be able to test this because we know what the assembly, we can get proxies of the measurements of assembly of individual molecules in the lab. And it's also connected to some of the network theory work that we were doing before. And so one of the sort of experimental systems we work with that Lee has in his lab now is called the computer, and they do these soup experiments where they have random small molecules and they subject them to different environments, which might be different minerals in, that are steering the chemistry. And there was a paper we were involved with their group several years ago, basically showing that you can't predict the microstate distribution, like the exact details of the chemistry in these experiments based on what the steering conditions were, but you can predict macro scale features of the product distribution. So if you think back to the biochemical systems I was talking about, we were interested in statistical patterns that were universal and could describe features of biochemistry, not the specific molecules. And what they're showing is when you do these prebiotic chemistry experiments and you're randomly exploring chemical space, but controlling sort of the environmental conditions, what you can predict is the macrostates, not the microstates. And that allows us some possible mapping between those theories. And the way that they're measuring that the system is acquiring information is by measuring the assembly of the assembly indexes of the molecules. And they've been able to show that certain conditions actually can increase the assembly number. So you're putting information into the system and you can see the evidence of that evolutionary process that you're inducing on the system as an agent in this increase in assembly. And so part of my goal with this is actually to try to think about origins of life in a planetary context. And sort of we have this vision that we're building with a bunch of other groups of building larger scale experiments that allow us to explore more of chemical space and start to look for life-like chemistries and try to think about the assembly space kind of ideas, the informational space of chemistry, and developing theory with people at SFI about that. And sort of my sort of pitch for why this needs to be done is just to you know, the physicist pitch. This is Super Kamiokande, which is one of my favorite experiments that's running right now. But for those of you in particle physics, you know that this experiment is trying to bound the probability of proton decay. So we basically built this massive experiment to look for an event that's never happened in the universe, but we can predict its likelihood of or like bound, you know, how probable it is. And most of the experiment has basically been increasing the lifetime of the proton over the duration of the fact that they haven't observed an event. Origins of life is a little bit different because people don't think about it as a probabilistic process that happens in our universe, and there's some like statistical constraints on that process that we need to get in order to talk about how likely we're going to be able to find it in a planetary environment off Earth, which is critical to designing what kind of telescopes we need to build. Should we build focused efforts that allow us to get a lot of detail from a single exoplanet if we think there's a high likelihood of life? Or should we build experiments that survey ensembles of exoplanets to extract as much information as possible because we think life is rarer in the universe? And we don't have any bounds on that. The proposal is sort of, we need to have more of these kind of experiments that are statistically exploring chemical space and looking for these kind of lifelike properties and try to map the theory to the experiment and actually build origins of life experiments that are asking these kind of fundamental physical questions about the nature of the transition from life to non-life. And I do have money now from NASA to build a geocomputer, which is a computer that's specifically coupled to planetary environmental context, which we're going to build at ASU as part of a recently a funded NASA project in the Interdisciplinary Consortia for Astrobiology Research. So I'm very excited about this, and we might start actually be able to do some of these things and mapping some of that biochemical network work and like some of the statistical patterns and some of these ideas about the increase in sort of the information in the molecules that biochemistry is using and things, and bring these ideas together to try to map out sort of like what kind of theory and experiments we need to do for origins of life. And I will leave on this quote because it's my favorite, because I feel like this every day. One can best feel in dealing with living systems how primitive physics still is. And then I just want to thank my group, who's really awesome, and all of our funders, and thank you guys for listening. And I'm happy to take more questions if there's time.